surrounding 160. All these potential phosphorylation sites, they are either phosphorated or dephosphorated and play an important role for regulating cyclin CDK activity. These phosphatase, particularly, is able to dephosphorate these surrounding 160 residue, as you can see here. Okay, this original phosphorylation on this residue, once got dephosphorated, and these cyclin CDK become inactivated. Another one is by adding the inhibitory phosphate onto an important residue, furan 14 and tyrosine 50. So this is mediated by WEE1/MYC1 kinase. The kinase add a phosphate on these two residues, and this cyclin CDK become inactivated. So four mechanisms. For biological mechanism to inactivate the cyclin CDK activity. So it's definitely much more complicated than our motorcycle, more complicated than our car system, right? For biochemical mechanism, don't you, I guess you don't need the four step or four way to start your motorcycle, right? You just step on the brake, that's it, right? So you could imagine this is much more complicated. So I just want to summarize that part, but I want to add another one, especially the first mechanism. Even the first mechanism, by associating with CDK inhibitor, is also not just simple CDK inhibitor associated. Among these CDK inhibitors, they are two families, and make it even much more delicate to control the cell cycle progression. Actually, the two CKI family, one is called INK4 family, the other one is called CYPKIT family. INK4 stands for inhibitor of CDK4, okay? Inhibitor of CDK4. They are including the several protein called P16, P15, P18, P19. We'll talk about that later. Their capability is to inhibit the early G1 cyclin CDK activity, okay? Thereby stopping the cell cycle machinery. The second family of CKI called CYPKIT family stands for CDK interaction protein or kinase interaction protein. They are including P21, P27, and P57. So their capability is to stop the cyclin eCDK2 activity, thereby causing cell cycle arrest. Okay. So the cell cycle machinery goes through the cell cycle clock, so goes through different phases of cell cycle. But they could be controlled by CKI, they could be controlled by CAP, they could be controlled by WEE1/MYC1 kinase, they could be controlled by cyclin degradation. But here is basically the first biochemical mechanism by associating with the CKI. So what is the CKI? CKI stands for CDK inhibitor. Okay? As I mentioned, two families. One is called INK4 family, stands for inhibitor of CDK4. The other one is called CYPKIT family. We have P21, P27, P57. We have P15, P16, P18, P19, etc. Okay? These are what we call the family because the protein structure, they share the same domain. For example, for the CYPKIT family, at the N terminal, they all have these CTK binding domain, okay? And then P21, P27, they are importantly induced by important signal. Okay, for example, P21 is induced by P53 or TGF-beta. P53 is the #1 tumor suppressor gene protein. Okay, it's important to know this particular tumor suppressor gene because P53 is highly mutated or deleted in more than 50% of cancer. Okay, you collect all the cancer, no matter it's from could be coming from ovarian cancer, coming from lung cancer, from other type of cancer. Okay? If you check the PPD3 status, PPD3 is highly mutated or deleted. Okay? So you could know, you could appreciate how important this important tumor suppressor gene. So this important tumor suppressor gene has a very important biological function, is to control the cell cycle. How it could control cell cycle? By induce the expression of P21, okay? And thereby causing the CDK inactivation, causing cell cycle arrest. Another one called P27, okay? This particular P27, just like P21, could be regulated, and then is involved in the differentiation process. P57 could be induced by TGF-beta. This is a very important cytokine. TGF-beta involved in the cell growth inhibition. Okay? TGF-beta, how TGF-beta could have that capability to do so is because it had a capability to induce P27 or PPT7. We'll talk about that later. The INK4 family, okay, the inducer, one of the inducer PPT is induced by TGF-beta. We will have chance to talk about 43 sigma. It's a very interesting protein. Actually, this is also involved in cell cycle regulation. Okay, and then it's also induced by very important tumor suppressor gene, P53. Okay. We'll spend some time working on this particular protein called 1433 sigma. So. I want to introduce you the INK4 family. INK4 family, how could they inactivate CDK4? Okay, you coming from the name called inhibitor of CDK4. Okay, now you see that all these INK4 family, basically they inhibit CDK4 is by competing with cyclin D. Okay, they will bind to CDK4 or CDK6 directly. The structural biologist has determined the structure between CDK4 and P16 based on this here. This is on the CDK6 and P16 binding. So they have direct binding. By doing so, they are competing with the cyclin. So when they bind here, the cyclin D level off, and then the CDK4 or CDK6 become inactivated. So this is one mechanism for the INK4 family. But for CKIP family, a little bit different. Okay? You look at the structural biology shown here. The cyclin and CDK. Okay? Cyclin associated with CDK, these CDK become activated. And they have the important site for the kinase substrate to enter. So this is CDK will be activated. For example, look at here, this is the ATP binding region. Okay. They have this pocket for the substrate to enter the CDK, and the CDK will have the capability to catalyze, to phosphorate the substrate. So they have this substrate entry site here. Interesting enough, P27, okay, was able to inhibit CDK activity by blocking that substrate entry site. You can see here, the P27 has these, based on this green structure, by blocking that substrate entry site. So the CDK cyclin complex become inactivated. Remember, this is different from INK4 mechanism, because INK4 was doing competition. Okay? Here is not doing competition. P27 is doing so by blocking the substrate entry site. Okay? It's different from INK4 family. And this is all based on the important study of the structural biologist. Here, look at again, P27, P21 is one family, INK4 family is another family. The INK4, we call it family because it has these so-called anchoring repeat. Okay? This is the protein interaction motif. So you could imagine that this is CDKI, the CKI. They are important, potentially, we call it break, okay? And then we think they are potentially tumor suppressor gene because the break is always carefully controlled to make sure the cancer cell will not going on without the break, right? So these break potentially they are the so-called tumor suppressor gene. So indeed, we do some study to understand whether they are mutated or deleted in human genome, in human cancer genome. So location of these different genes has been studied. They have been mapped into, for example, P27 mapped to chromosome 12, P12, the P arm of the chromosome. P21, 6 P21, PPD7, 11, P15.5. Okay. P16, P15, 6. They are also been mapped. This is the chromosome location. 1 P32 for P18, 19 P13 for P19. Interesting enough, for these families, actually, the mutation found in cancer is so rare. So we've been scratching our head for a while. But, you know, if the break is so important, and maybe the gene has some mutations, so the cancer cell will have the advantage to inactivate the break. But actually, the defect actually is not at the gene level. Okay, it's actually at the protein level. Okay, I will tell you the story how the cancer cell has the capability to inactivate the protein's biological function. Okay, so this is just to share with you. But another type of family, P16, P15, this one does has a gene defect. Okay, for example, the 9P21 and 9P for the P16 and P15, they have a deletion in the cancer cell line, plus some of the melanoma cell, human melanoma. So indeed, this is sort of kind of like a typical tumor suppressor gene. And here to summarize some of the study about all these biological activity of the INK4 family or SIPKIP family, plus some of the important regulation of these particular genes. Okay. For example, P16, we know that positive regulated could be coming from the transcription factor. The P16 could be coming from the suppression due to the activation of MYC oncogene, due to the promoter methylation, due to the deletion or mutation on this particular gene. P16 could do several things. Okay? They could cause replicative senescence called anokia. P16 be able to suppress the angiogenesis, it suppresses cell spreading, suppresses telomerase activity, suppress the cancer growth, suppress VEGF expression. So you could imagine if a cancer cell managed to inactivate these particular genes, all these capabilities become activated. Those cancer cell will have the capability to do new blood vessel formation, will be able to do cell invasion, spreading out, be able to deregulate telomerase, activate more telomerase, be able to promote cell growth, be able to promote VEGF production, etc. You could imagine that why these genes need to be inactivated in cancer, so the cancer cell will have the growth advantage, right? So the P15 almost do a very similar thing like P16. It's been suppressed by MYC, suppressed by promoter methylation, and will do other things. As for P21 and P27 and PPD7, they've been regulated in different pathways as well. P21 being regulated by PPD3, as I said earlier. It could be regulated by TGF-beta, And then it could be suppressed by some of the important oncogene. The oncogene like MYC or HER2. HER2 stands for human estrogen receptor type 2. Is very important receptor tyrosine kinase. You will be able to regulate, suppress P21 expression. P27 is very similar situation, and it's been regulated by several important tumor suppressor genes. Okay, the P10, FKHR, the forehead transcription factor, VHL, a very important E3 ligase involved in tumor suppressing activity. They will be able to promote the P27 expression. The oncogene, the HER2 MYC, promoter methylation of JEB1, suppress P27. So you could see the cancer cell have different mechanisms to cause these inactivation of CYP family or INK4 family. P57 almost doing the same thing, like the P27. It's been regulated by TGF-beta, been suppressed by IGF-2, okay, promoter methylation, and then another way is called genomic imprinting. Genomic imprinting was able to suppress the P57 expression. It's worthwhile to mention that P57 actually is paternally imprinted, meaning that you and I, many of us, only one copy of P57 gene is expressed, and it's coming from your mother, not coming from your father. Okay, it's so-called genomic imprinting. So it's paternally allele has been inactivated. Okay, so that process is called genomic imprinting. So many of us only have one copy of a P57 gene expressed. Again, we want to understand the biological function of these CKI, and many knockout strategies has been used to understand more the biological function of these important CKI. So knockout P21, P27 has been done. But interestingly enough, just like some of the CDK knockout, P21 knockout, no growth defect. Okay? Again, suggests P21's biological function could be replaced. Okay? P27 knockout, again, no growth defect. Okay? Except P57. Okay? P57 knockout has so many defects. Okay? They have a cleft palate, they have a gastrointestinal tract defect, sometimes they do not have the intestine, sometimes they do not have stomach, okay, once you knock it out. Okay? And then they have a defect in the body weight, they have the adrenal cortex enlargement, they have renal medulla dysplasia, they have an eyes problem as well. Okay? So imagine P57 even much more delicate to be controlled because we only have one allele from our mother. Okay? Mouse is the same thing. Mouse only is paternally imprinted, so only the maternal copy is expressed. But once P57 got lost, they cause all these different defects. So this study allows you to have a better idea about how these important cell cycle inhibitors, how they are regulated. Right now we want to go into one side of how scientists start working on these CDK inhibitors. Okay? I personally had the experience in doing some of these studies, and it was a great experience for myself. And I would like to share with you the P27 was cloned using a biochemical way, and then to clone the gene and allow us to understand. And I had sequenced all the P27 genes. Okay. If you go to the database, maybe you could still see my name there. Okay. But just to share with you how that was done there, we prepared the cell extract from the growth arrested the cell, and then go through the affinity column. Okay. This affinity column have the cyclin E-CDK2 complex there, because we are looking for cyclin E-CDK2 associated protein. So we use an affinity column. Okay? This affinity column is composed of the cyclin ECDK2. So we pass through this affinity column. All molecule, potential molecule, microcyclin ECDK2 will be stuck there. Right? And then you elute it out, okay? And then, of course, CDKO2 will be eluted out, cyclin E will be eluted out, including P27. And then we run a gel, purify the protein band, and get the protein sequence by doing the so-called microsequencing. And we'll get the peptide sequence. Based on the peptide sequence, we design an oligoprimer that could encode the P27 protein. And using this primer to screen a cDNA library, many of you probably don't know what a cDNA library is. And then you have to pull out clone the gene and then sequence it. So it was a very laborious process during that time. But that's sort of a story to share with you, how human knowledge will allow us to be able to tackle all these problems, be able to understand more of the gene involved in this process. And then later on, several groups all study all these CKIP families. So later on, we have given the nomenclature showing that these are cyclin CDK, indeed, inhibitor has a family, has a common feature. The common feature, as you can see here, we made up all the sequence of P21, P27, PBD7, and you see the C-term at the N-terminal part seems to have some homology, okay? And then the C-terminal part has some homology as well. But the major homology is located here, and these happen to be the so-called CDK binding domain. If you remember a couple slides before about the protein feature of the CKI family, they have the CDK interaction, CDK binding domain. It's actually located in the N-terminal of P21, of P27, and PBD7. So how does it cause a cell cycle arrest? By interacting with the cyclin CDK, it directly inhibits the cyclin CDK activity. And I want to show you how scientists could prove that causing cell cycle arrest. It was done by the so-called FACS analysis, stands for flow cytometer activating cell sorting, okay? FACS, okay? Flow cytometer activating sorting, cell sorting, okay? And this allows you to, based on the DNA content, to understand the cell cycle proliferation process. So these are the y-axis and the x-axis. The y-axis meaning the cell number in solid light. The x-axis meaning the DNA content. Okay. The first peak usually is 2N genome content. The second peak is the 4N genome content, meaning that these cells already finished DNA synthesis, right? Because you have to finish DNA synthesis in order to be a 4N content. In between, that was called the S phase, because during this process, the cell is duplicating their genome from 2N to 4N. So between this peak, we call it S phase. So the first peak is G1 phase, which is 2N DNA content. In between is S phase, because DNA undergoing DNA synthesis. The second peak is 4N content. We call this peak as a cell located at M phase or G2M phase. Okay. So based on this, when you read the literature, you will know the first peak is G1 phase. The second peak is G2M phase. In between, that's S phase. Okay. Now we express the KIP2 of P27. P57 and P27 into the cell and do the cell sorting. Right now you see only one peak show up, but not second peak, right? So only one peak show up, we call it G1 arrest, because P27 and PPD7 was able to stop the cell from progression into the DNA synthesis or going to the mitosis phase, okay? So based on these, then you have the percentage calculated here. The control, okay, has a normal cell proliferation profile. You see some G1 peak, you see some S phase, you see some G2M phase. But the KIP1, P27, KIP2, PPD7, there's a high percentage of cell located G1, okay, meaning that they were able to stop the cell cycle. Okay. So this is a very important observation. So here, this slide here, to summarize all the one I just mentioned to you, a cell cycle machinery. You just imagine every day you're riding a motorcycle. You control your motorcycle. But inside your body, your cell, every single cell, are doing the cycling for you, okay? They have to go through all these processes, G1S, G2M, and the early G1 is controlled by cyclin CDK complex, but this cyclin CDK complex have to be controlled by a brake system. For example,